I wish I had this sort of video when I was studying for the MCAT or even for my biochemistry class. In this video, we're going to cover everything you need to know related to amino acids when it comes to the MCAT, the DAT, or even your biochemistry course. So we're going to cover things like side chains, broad categories of amino acids like hydrophilic, hydrophobic. We're going to cover acidic versus basic amino acids. We're going to cover the pi value or the isoelectric point. We're also going to cover R and S absolute configuration and L/D classifications and more. So this is going to be a long video. You may want to watch it over multiple sittings or maybe just one sitting and watch it multiple times because this is literally the highest yield topic on the MCAT. Also, in the description box, you'll find an Anki deck specifically for amino acids covering every single concept we cover in this video to aid with memorization, as well as a practice sheet to help you memorize the side chains of the amino acids. So without further ado, let's get started with our discussion on side chains, and I'm really going to relate the side chains to one another to aid with the memorization process. Before we get started, if you haven't yet subscribed to my channel, be sure to like, subscribe, and hit that notification bell to stay up to date with all of my content. Also, share these videos with your friends. It really helps the channel grow, and I would really appreciate it. And also, I think your friends will appreciate it because we're going to discuss a ton of high-yield MCAT content and biochemistry content. Also, if you want to support the channel, check out the links in the description box, the Patreon link or the Buy Me a Coffee link. But of course, support, while appreciated, is never expected. All right, let's jump into the video. So it all centers around this central alpha carbon. So this carbon, we call it the alpha carbon. It's attached to 3 groups. In reality, you have 4 groups, but we're going to omit hydrogen. So one of them is going to be hydrogen. All right, so over here we have our carboxylic acid. Here we have our amine, and finally here you have your R group. Now, of course, what's going to make the different amino acids unique is this R group right here. So we're going to be changing this R group, and that basically represents the side chains of amino acids. So the very first amino acid that we're going to discuss is going to be glycine, the most simple one, abbreviated GLY. One letter code is G. Remember, you need to memorize both of these, the three letter abbreviation and the 1-letter code. Now, the side chain of glycine is simply hydrogen. Now, of course, we mentioned earlier that there was also another group over here, which is also hydrogen. So it turns out that glycine will have 2 hydrogen atoms attached to the central alpha carbon. And for this reason, glycine is the only amino acid that is going to be achiral. Okay, so it doesn't have an absolute configuration or like the L/D classification because it has 2 hydrogen atoms over there. All right, so now let's move on to the next amino acid. Also pretty straightforward. The difference here is that instead of having a hydrogen atom, you're going to have CH3 or a methyl group in its side chain. And this one's going to be alanine. Okay, and alanine is abbreviated ALA, and the 1-letter code is A. Pretty straightforward. Now, glycine and alanine and the next several amino acids we're going to discuss are hydrophobic. Now, they're not all equally hydrophobic, as we'll see. Some of them are more hydrophobic, some of them are less, but nevertheless, they are all hydrophobic, at least the next several ones we're going to mention. So now let's move on to the next amino acid, and notice I'm going to relate this side chain to the next amino acid, which we are going to discuss, which is valine. Okay, valine, abbreviated VAL, and then the 1-letter code is V. Now, notice the difference over here. So you keep that methyl group, or at least the carbon. So we still have a carbon after this alpha carbon, and then directly after that, you're going to draw this V. Okay, and this makes sense. You have V in the name of valine and in the side chain. So that is the side chain of valine, and that's how you will remember it. So whenever you see an amino acid that has a V that's coming off directly from this alpha carbon, that is valine. All right, now let's move on to the next amino acid, which has a very similar side chain to valine. Again, because valine has hydrocarbons in its side chain, it only has carbons and hydrogens, it is non-polar. So the next amino acid we want to discuss is going to be leucine. Okay, we'll start with leucine over here, abbreviated LEU, and then the 1-letter code is L. Now, notice over here, we're going to have a V in the side chain as well, but it's not coming off directly from this central alpha carbon. You're going to have one carbon over here, and then you have a second carbon, and then off of this second carbon, that is where you have your V coming off. So the difference between leucine and valine is that the V comes off 2 carbons after the central alpha carbon. So you have a carbon number 1, carbon number 2, and that's where your V comes off from. And again, because these are hydrocarbons, this is a hydrophobic amino acid. All right, so really quickly, actually we'll discuss what are like some sort of questions that can come up related to hydrophobic amino acids once we're done discussing the hydrophobic amino acids. All right, now we'll talk about isoleucine, and notice it's probably going to be similar to leucine, right? And that's why we call it isoleucine. Okay, and its 3-letter abbreviation is ILE, and then the 1-letter code is I. And remember, guys, I have that practice sheet in the description box down below. It's basically you walking through these on like a daily basis when you're studying for the MCAT. So it'll allow you to draw the side chains, the 3-letter abbreviations, and the 1-letter codes. Okay, so now the difference, now this here is still leucine. Isoleucine is going to look like this. So you're going to erase this methyl group over here, and instead of, so essentially you have 1, 2, and 3 carbons, right? Instead of the methyl group branching off at carbon number 2, it's going to branch off on carbon number 1. Okay, so in a way you can think of it in terms of like leucine has a V in its side chain, whereas isoleucine has sort of like a U in its side chain. Okay, so 1, 2, 3 carbons after that central alpha carbon, and then the methyl group is going to come off carbon number 1. Okay, next let's move on to, we're still going to discuss hydrophobic amino acids, but the ones we're going to discuss after this point are going to be aromatic at the same time. So when it comes to the hydrophobic amino acids, that is the non-polar amino acids, you have aliphatic. Aliphatic just means that it has hydrocarbon chains in its side chain, which is what we were discussing so far. Now we're going to discuss hydrophobic that are also aromatic. Okay, so we'll see what that means right now. So the first one we're going to discuss is phenylalanine, abbreviated PHE, and the 1-letter code is F. Now, the side chain is going to look like this. So after that central alpha carbon, you have 1 carbon, and then after that first carbon, you will have your ringed structure over here. So you can see that you have, and we actually call this a phenyl group, right? So like this structure would have been called a benzene molecule, right? Well, because you remove 1 hydrogen to be able to attach the rest of the amino acid over here, now you call this a phenyl group. Now notice the naming over here. Remember the structure of alanine? So the structure of alanine, let's just say that this is the carboxyl group, this is the amino group, and this is the R group. The R group was simply a methyl group over here. So now you're just adding that phenyl group, and that's why we call it phenylalanine. So I don't know, that just helps with memorizing the structure of things if you understand the breakdown of their names. Okay, now let's move on to the next aromatic amino acid. And I'll tell you what's unique about aromatic amino acids in just a second when I'm done discussing them. The next one we have over here is going to be very similar to phenylalanine. The only difference is that you're going to add an OH over here. Okay, when you add this OH, now this amino acid is known as tyrosine. Okay, and tyrosine is abbreviated TYR, and the 1-letter code is Y. Now, again, this is aromatic, so there's something unique about that, but there's also one specific thing that's unique about this specific aromatic amino acid, and that's the fact that it has this OH. Whenever you see OH in the side chain of an amino acid, and especially if it's not part of a carboxylic acid, because we'll see some amino acids have an OH, but it's part of like a carboxylic acid. But when you see OH, such as in this case with tyrosine, it can be phosphorylated. Now, what does this mean? Essentially, after a protein is translated, so after you have the amino acids join one another, you can have what's called post-translational modification, so modification to that peptide, to that polypeptide. One of the forms of modification is that you can phosphorylate this, so like you add a phosphate group. And remember, phosphate groups carry negative charges. So this can impact potentially like the signaling of the protein, where the protein is going to end up, the function of the protein. So just know that it can be phosphorylated because this is very high yield. And we're going to see more amino acids that can be phosphorylated as well. Now, importantly, while this is aromatic, and the fact that it's aromatic is going to make it hydrophobic, so that's why we generally classify it with the hydrophobic amino acids. Because it has this OH, it has some capacity to participate in hydrogen bonding, so it's going to be more at least less hydrophobic than some of the other hydrophobic amino acids, such as phenylalanine, or the next one we'll see, which is tryptophan. So just keep that in mind, that there are varying degrees to the level of hydrophobicity of these amino acids. Okay, let's move on to the final aromatic amino acid, and then I'm going to tell you what's unique about aromatic amino acids. Okay, so let's not screw this up. We need to draw a 5-membered ring. Okay, that's 5. 1, 2, 3, 4, 5. Okay, and then a 6-membered ring. Okay, all right, now we have benzene over here. Of course, this is tryptophan, abbreviated TRP, and the 1-letter code is W. What's unique about the 3 aromatic amino acids that we discussed, so tryptophan, phenylalanine, and tyrosine? I want you to always remember 280. What do I mean by this? Well, 280 nanometers is the ultraviolet wave that they will absorb if you try to like analyze them with UV-vis spectroscopy. So the graph that you'll see on test day will look something like this. You have the wavelength, like lamina over here, and this will be like absorption. And so what you'll see is, like, let's say this is 280. You'll see a peak at 280 if your protein sample contains the aromatic amino acids. So the way they'll ask you about this on test day, so you have 2 protein samples, you run them both using a UV-vis spectrophotometer, and you see that one gives you a peak at 280, the other doesn't. What does this tell you about the protein that gives you that peak? Well, you know that it probably has an abundance of these aromatic amino acids. All right, now let's move on. We're not done with the hydrophobic amino acids, but these are a bit more unique, so I wanted to leave them for the end. So the next one we'll talk about is going to be methionine. Methionine. I think I spelled that right. Luckily on the MCAT, you don't have to spell it right, because you don't even have to spell it at all. All right, M-E-T, you should know the 3-letter abbreviation and the 1-letter code, which is M. Now, what's unique about this is that it contains a sulfur group in its side chain. Now, it's not the only amino acid. We'll see cysteine also has a sulfur in its side chain. So the way we draw it, or at least recognize it on test date, is you have after that alpha carbon, you have 1 carbon, 2 carbons, and then you go to your sulfur, and then finally a methyl group. So this is what it looks like. Now, a confusing point here is, well, when we talk about cysteine, we're going to see that cysteine is capable of forming disulfide bonds because it has sulfur in its side chain. And that's only because in cysteine, as we'll see, sulfur is the last thing. Like, the last thing is you have an SH group, so it's capable of doing that. Now here, you can see that this isn't a terminal sulfur group. Like, after the sulfur group, you still have another carbon atom, so it's not capable of forming disulfide bonds. Nevertheless, sometimes they'll ask you about the fact that methionine has a sulfur group, so you should know that it does contain a sulfur group in its side chain, or a sulfur atom in its side chain. Okay, let's move on to, I believe this is the final hydrophobic amino acid. And this one is quite unique. I'm first going to draw it kind of the incorrect way to draw it. So you have, after the central alpha carbon, you have 1 carbon, 2 carbon, and 3 carbons. Okay, now this is proline. I know this isn't the right way to draw it, and I'll show you why in just a bit, and why I drew it this way. Proline, abbreviated PRO, 1-letter code is P. Now, the reason why you won't see it like this in your textbook or even on test day is because it turns out that this carbon is going to interact with the amino group the amine over there. And don't ask me how or what type of reaction occurs. I'm not an organic chemist. I'm here to teach you biochemistry. And even in the context of organic chemistry for the MCAT, I think this is beyond the scope. In either case, this reacts with this. So now what happens is you do have a ring in the side chain of proline, but technically that ring is forming between an interaction between the side chain and the amino group. So you won't see an amino group in the backbone because it's incorporated within the ring. So what it will actually look like is something like this. So you will have this. So you have 1, 2, 3. I want to make sure I don't screw this up. I always draw, when I'm trying to draw a pentagon, I draw a hexagon. So yeah, 1, 2, 3, 4, 5. Okay, and then of course you'll have an amino group right here. So you have NH. Okay, so very important. If you see it, like if you see this as an answer choice, obviously it's the correct one. If you also see something like this, where you have the backbone amino group, this is wrong. They're gonna try to trick you. You're gonna say, yeah, well, every amino acid has an amino group in its backbone and a carboxylic acid group in its backbone, and so this is probably the correct answer. But in reality, you should not see, for proline specifically, you should not see an amino group backbone. All right, so now before we move on to the next group of amino acids, the hydrophilic or the polar amino acids, specifically the polar uncharged ones, let's go over a few different questions that can pop up on test day, and I'm just gonna verbally mention them. So they can ask you, where would you likely find amino acids, hydrophobic amino acids? You'll find them in the core of globular proteins, because that way they're away from the aqueous environment of the cell, because they don't want to interact with the aqueous environment. You can also find them embedded within the core of the plasma membrane. So like interacting with the fatty acids, of course, you wouldn't find them necessarily interacting with the hydrophilic heads, because obviously they're hydrophobic. They can also ask you things like about some specific ones. So as we mentioned, methionine has sulfur in its side chain, but it's not capable of forming disulfide bonds. Also, the fact that proline, because of the structure we discussed, the unique structure, is going to sort of introduce kinks into alpha helices. Alpha helices are a type of one of the secondary structures of proteins, so they'll introduce kinks in there. Glycine can also introduce kinks because its side chain is really small, so it just contains a hydrogen atom. All right, so now let's move on to talk about the hydrophilic or polar uncharged amino acids. So the first one we're going to discuss is going to be serine. Serine is pretty straightforward. What you'll see is you have your alpha carbon, you have one carbon coming off of that, and then off of that carbon you're going to see an OH. All right, so this is serine, abbreviated SER, and the one letter code is S. Now again, because you have an OH over here, again, we said whenever you see an OH that's not part of a carboxylic acid in the side chain, the amino acid is going to be capable of being phosphorylated, right? So you label it with a phosphate group, and the phosphate group carries a negative charge, which has all types of implications. The most important thing is that you know that this is a form of post-translational modification that occurs within cells, and it plays a role in signaling, activity of the enzyme or the protein, etc. Okay, let's move on to the next amino acid that contains an OH in its side chain. So remember, we first mentioned tyrosine, contains an OH in its side chain, serine. The final one that contains an OH in its side chain is threonine. Okay, so threonine is going to be very similar to serine. So remember, in serine you had the first carbon after the alpha carbon, and then directly off of that you had the OH. So it's kind of the same thing. You have an OH. The only difference is that you add a methyl group over here. So now this is the structure of threonine, abbreviated THR. One letter code is T. That's why tryptophan's one letter code is W, because T was taken up by threonine. Okay, so this is the third and final amino acid that contains OH, which can be tagged with a phosphate group. Now, this isn't the final hydrophilic amino acid. We're still going to discuss a bunch of those. It's just the last one that has OH in its side chain. OH, by the way, that is not part of a carboxylic acid group, because as we'll see, some amino acids have a carboxylic acid in their side chain, which obviously contains an OH, but are generally not phosphorylated. Okay, so now let's move on to the next two, the final two hydrophilic or polar uncharged amino acids. So the first one we'll discuss is going to be asparagine. Asparagine, yes. ASN. And the one-letter code is N. So the structure of asparagine is going to look like this. You have your first carbon after the central alpha carbon, and coming off of that is going to be your, what we call an amide group. So an amide group, remember, is a carbonyl with an amino group. Now, importantly, this cannot be deprotonated or protonated at physiologic pH. A lot of times students will see NH2 over here and think that asparagine, and as we'll see, glutamine, can carry a positive charge if phosphorylated, if protonated. That's not the case. This cannot be protonated, and therefore it is polar, okay, because it contains an amide, but at the same time it is uncharged, okay? So we'll move on next to glutamine. Glutamine. Okay, abbreviated GLN. One letter code is Q, right? Because G was taken up by glycine. Okay, the only difference here is, I'm actually not going to redraw the whole thing. I'm just going to introduce another carbon atom. So the only difference is that you have, well, okay, let's do this. So I'll erase this. You saw how you had one carbon after the central alpha carbon. Here you have 1, 2. Okay, so you have one carbon, 2 carbons, and then you have your carbonyl and your amine, which is, your amide. So very similar to the structure of asparagine. The only difference is one carbon after that central alpha carbon. Again, this is polar, uncharged. Now we're going to move on to the polar charged amino acids. We're going to start with the acidic ones, and then we'll discuss the basic ones. Very quickly, before we move on to the polar charged ones, I did want to mention a few things, a few type of questions they can ask you on test day. So they can ask you, obviously, where will you find the polar amino acids? You'll find them on the surface of globular proteins. You can also find them interacting with the hydrophilic heads in the phospholipid bilayer. Also, specifically for the ones that contain OH in their side chain, very high yield to know. Again, I can't emphasize this enough, that they can be phosphorylated. Okay, I believe that's it. That's all I wanted to mention for the hydrophilic uncharged ones. So now let's move on to the hydrophilic charged ones. And as I said, we're going to discuss first the acidic ones. Now, of course, the acidic ones are the ones that can carry a negative charge, right? Because they're acidic, they can donate a proton. When you lose a proton, you carry a negative charge. So the first one we're going to talk about here, and there are only two of them, is aspartic acid. Aspartic acid, sometimes called aspartate, and we'll see, why does it have like two names? I'll explain why in just a bit. Aspartic acid, abbreviated ASP, one letter code is D. Okay, and then what you have here, very similar to what we saw with asparagine, you have your first carbon after your central alpha carbon, and then you have your carbonyl group coming after that, so you have your carbonyl, and then an OH. So this is, of course, a carboxylic acid. Now, because you have a carboxylic acid, again, this is carboxylic acids are acidic, so at physiologic pH, they can donate a proton. When you lose a proton, you carry a negative charge, and that's why the acidic amino acids are negatively charged at physiologic pH. Now, we're going to go in more detail of like when will this be protonated and this be protonated and the PI value after we discuss all the side chains. So don't worry about that for now, okay? As I mentioned earlier, aspartic acid, sometimes you'll see it as aspartate. Aspartate is essentially the nomenclature for the conjugate base of carboxylic acid. So this is a carboxylic acid, that's why we call aspartic acid. Once you donate the proton, then it's called aspartate. So that's where the naming comes from. Notice we had asparagine. The only difference between asparagine and aspartic acid looked like this. So asparagine looked like this, right? So you had carbonyl over here attached to an amine group. So this was an amide. So notice the only difference is that instead of having an amide in the asparagine, in the case of aspartic acid, you have a carboxylic acid. Now, same exact idea. You know how we discussed, so in asparagine, you only had one carbon atom after that essential alpha carbon, and then in glutamine, you had 2 carbon atoms, right? Same exact idea. If you want to change this structure and make it glutamic acid instead, abbreviated GLU, one letter code is E. Now what you do is you just add one more carbon. So here we had 1, 2, so 1, 2, and then your carboxylic acid group. Now, a few things they can ask you about the acidic amino acids. First of all, you want to know that they're going to carry a negative charge. So sometimes they'll give you like polypeptide chains, and the polypeptide chain, and typically they'll give you them in like the 1-letter codes. So it'll be like A, T, E, D, L. So, and then they'll ask you like which one's going to migrate farthest on gel electrophoresis. So I can go through specific examples in future questions, but I want to in future videos, but I'll give you kind of a general idea of how they ask the questions. So over here, what you want to know is that this polypeptide is going to carry an overall charge of -2. How do I know that? Because alanine doesn't contain a charge. T, which we saw earlier, is threonine, doesn't contain a charge. It's neutral. E, which is glutamic acid, is -1, and D, which is aspartic acid, is -1, and L, which is leucine, is neutral. So the overall charge here is -2. So they ask you about that. They can also ask you, so you'll find that both the acidic and the basic amino acids can sometimes be found at the active sites of enzymes, and that's because they can either accept or donate a proton, and that kind of alters the local environment. So you can find it at the active site of enzymes. And I believe that is it. Of course, there's a lot more to talk about when we talk about titration curves or the PI values of amino acids. So we'll get to that when we're done discussing the side chains. Okay, so, and then notice one very important thing that a lot of students screw up. So not only do, like for example, this was the structure of glutamine, and this is glutamic acid. Not only will students often screw up that this, they'll think that this can carry a positive or negative charge because it has an H2. So they'll also screw it up in another way where they'll see GLU and GLN very close, you know, it's just that last letter changed. So they'll see GLN and they'll think that that's going to carry a positive or negative charge. So make sure that you memorize these really well, that glutamic acid is GLU and glutamine is GLN, which is the one that's polar but uncharged. All right, so now let's move on to the basic amino acids. We're coming at the home stretch over here, guys. So the basic amino acids, we have 3 more amino acids. Well, after we're done with the basic amino acids, we just have 1 more amino acid. I left it for the end because it's quite unique, which is cysteine. A lot to talk about when it comes to cysteine. For the first basic amino acid is going to be lysine, okay, and it's going to contain 4 carbon atoms after that central alpha carbon. So 1, 2, 3, 4. And then after the 4th carbon, you have NH2. And this right here is what's capable of being protonated. So when you protonate this, it's going to carry an overall positive charge, okay? Again, we're going to discuss like when this one will be deprotonated versus this one when we talk about the PI values of amino acids later in this video. So this is the first basic amino acid, lysine. And then the second one that we're going to discuss here is arginine. Arginine, okay, ARG. And then the 1-letter code for arginine is going to be R. And this one has a pretty unique side chain. So you have 1, 2, 3. As you can see, I always count the number of carbons because I always screw this up if I don't like diligently count them. So what it looks like is something like this. So we have a single bond to NH, and then off of this NH, we have a single bond to carbon, and then you have this carbon nitrogen double bond, and then finally your NH2. This amine over here that can be protonated, so if it's protonated, it carries an overall positive charge, and that's why it is a basic amino acid. And then the final basic amino acid that we're going to talk about is what we call histidine. Histidine, abbreviated HIS. One letter code is H. Okay, so this one is going to have a ring in its side chain. So notice you have a 5-membered ring, so 1, 2, 3, 4, 5. And notice you have 2 amines in it. This one's the one that can be protonated, so it can carry a positive charge, and that's why we consider this to be a basic amino acid, or a positively charged, or at least one that's capable of carrying a positive charge. Now this one is a bit unique because the pKa, and we're going to discuss the pKa of the individual groups, so the amine, the carboxyl group in the backbones, and the amine or carboxylic acid groups in the side chains in just a bit. But for this one, just know that the pKa will be about 6. Now that's pretty close to physiologic pH, so the overall pI of histidine turns out to be very close to physiologic pH. So this one, it's quite unique in that it's not necessarily going to be positively charged at physiologic pH. So for the purposes of the MCAT, I would really just consider lysine, which we abbreviated as K, and arginine, which we abbreviated as R, as the ones that are going to be positively charged at physiologic pH. And then the ones that are negatively charged are going to be D, which we call as aspartic acid, and E, which is glutamic acid. Histidine, I wouldn't really, in questions, I wouldn't consider it to be positively charged. Just know that it's capable of carrying a positive charge. Okay, so for cysteine, you can relate it to serine. So remember serine, and they both kind of start with, obviously they don't both start with S. Serine starts with S, cysteine starts with C, but they sound alike. So you can relate them to one another. So remember when we talked about serine, that one had the first carbon after that central alpha carbon, and then an OH. Well, when it comes to cysteine, I think I spelled that right, CYS, one letter code is C. Instead of having OH, you're going to have SH. Now that is very unique because if you have 2 cysteine molecules, let me draw this real quick. So this is going to be our central alpha carbon over here, and here is our other SH, let's erase this. Okay, so here is my SH. Okay, so these are both cysteine residues, and let's say they're in the same protein, either within the same polypeptide, within the same subunit, or different subunits. But essentially what they can do is they can interact with one another, and what you'll see is, and this is very high yield on the MCAT, you can form a bond between these. Okay, so you remove the hydrogen atoms and form a disulfide bond. Okay, disulfide bond. Now it's not enough to know that they form a disulfide bond. You want to know what sort of reaction allowed them to form that disulfide bond. Remember, I just said you remove the hydrogen atoms. Well, when you remove hydrogen atoms, what kind of reaction is that? Dehydrogenation? No. It is a reduction. No, it's an oxidation, right? Oxidation. What you're doing is you're oxidizing the cysteine residues, right? Because you had SH and SH. Now, remember, in the context of Gen Chem, we tend to think about oxidation and reduction in terms of gain or loss of electrons. In the context of orgo or biochemistry, think of it in terms of loss of hydrogen atoms. So if you lose hydrogen atoms, you're oxidized. If you gain hydrogen atoms, you're reduced. Or in terms of bonds to oxygen. So if you gain bonds to oxygen, so you go from a single bond with oxygen to a double bond, you are oxidized. And if you go the other way around, you're reduced. Here we're specifically focusing on hydrogen. So if I want to form the disulfide bond, what I do is I oxidize the cysteine residues and form that disulfide bond. Now what if I want to reverse this process? Well, you can reverse it obviously in reduction, in a reduction reaction. You'll need a reducing agent such as beta-mercaptopurine, and it'll allow you to break these disulfide bonds and form your cysteine residue once again. This is very high yield. Like you'll see questions related to electrophoresis, for example, where you'll have, for example, insulin is composed of two subunits that are joined to one another via disulfide bond. So they'll ask you, they'll have like on electrophoresis, obviously proteins are separated based on size, right? So their molecular weight. So sometimes they'll show you a sample that previously contained insulin, but is now showing multiple or two subunits on the electrophoresis, and they'll ask you what, like two bands on electrophoresis, whereas you should have had only one, right? Because you had one protein. What resulted in the fragmentation or the breakdown into two separate bands? Well, you probably added some sort of reducing agent and broke down those disulfide bonds, and now you have two separate subunits that were shown on the electrophoresis as two separate bands. All right, so there's one more thing that's unique about cysteine, but I won't mention it just yet because it relates to absolute configuration. So we'll talk about that when we talk about when we talk about how we assign the absolute configuration and the L/S classification. But for now, let's move on to PI values of proteins, okay? PI values of amino acids. So the PI, short for isoelectric point, okay? So the idea is that within an amino acid, let's go back to our generic amino acid, you have 4 groups, right? So the first group is going to be your carboxylic acid group right here in the backbone, amino group in the backbone, the R group, and the hydrogen, which we're not going to draw out. So for all of the amino acids, with the exception of 5, remember the 2 acidic ones, glutamic acid and aspartic acid, and the 3 basic ones, which are histidine, arginine, and lysine, all the rest of the amino acids will have a neutral side chain. So they are not capable of carrying a positive or negative charge, right? So that means the only 2 ionizable groups are going to be the amino group and the carboxylic acid group. Now, what will the carboxylic acid group can carry a negative charge, and the amino group can carry a positive charge, right? Because you deprotonate the carboxylic acid group, and the amino group can be protonated to carry a positive charge. Now, when the amino acid looks like this, we call it a Zwitter ion. What does it mean to be a Zwitter ion? It means that the molecule is neutral, okay? So neutral, but it's not enough, that's not enough to define a Zwitter ion because there are tons of neutral molecules. So for example, this is, I don't know, like methane. Is it a Zwitter ion? No, but it is neutral. But the idea is that you have to have both positive and negative charge on the molecule, canceling each other out and making the molecule neutral. Now, when an amino acid is in the form of a Zwitter ion, that is when its carboxyl group is negatively charged and its amino group is positively charged, and this is specifically for the ones that have a neutral side chain, this is the point where you're at your PI value or your isoelectric point. Now, we'll talk about the specific numbers in just a bit. The reason why we call it the isoelectric point is because it's the point where the amino acid will stop migrating on isoelectric focusing. Now, I don't expect you to know the details of isoelectric focusing from this video. I'll go in more details in a separate video. I actually already kind of discussed this at length in my lab techniques playlist, specifically in the 3rd video in that playlist. I'll have it in the description box down below. But just know for now that that is the point where the amino acid, because it's neutral, it'll stop migrating. So it's not going to be attracted by the negative or positive electrode on isoelectric focusing. All right, now let's go over the specific numbers. So first we'll do the specific numbers for like the pKa values for neutral amino acids, and then we'll talk about the acidic and the basic ones. So the way you calculate the PI value, because sometimes you will be asked to calculate the PI value or to give the rough estimate of the PI value, is that you want to take pKa1 plus pKa2 over 2. So very simple, you're just averaging the pKa values. Okay, so that's going to equal your PI. What is pKa1? This is the pKa of your carboxyl group. pKa2 is the pKa of your amino group. This is going to be approximately 2.5 to 3. It doesn't really, like, you don't have to know the exact pKa value. Just know that it's roughly like 2.5, that it's more on the acidic side, okay? And then the pKa2 over here, because it's an amino group, is going to be closer to like 9, okay? So if you were to take the average of that, 9 plus 2.5 is 11.5, and just so that this example sort of works out, let's just round it up to, say, 12. When you divide by 2, okay, when you divide by 2, you get 6. And that is indeed the pi value for the neutral amino acids. It's going to be about 6. Now, is it exactly 6 for every single one? No. You might see 5.7, 5.8, 5.9, 6, 6.1, so on. But you get the point. The point is that the pi value is around 6. Okay? So at physiologic pH, which is very close, 7.4, they're gonna carry an overall neutral charge. The majority of the amino acids will be in this zwitterion form. Okay, now let's move on to how to calculate the pi values of acidic amino acids. I hope my handwriting, I know, is horrible, but since I'm like kind of reading things through as I'm going, I hope everything is clear. Okay, now you can see why it becomes it's kind of a unique situation when you have a side chain that can carry a positive or negative charge. So let's just go with aspartic acid over here. So remember, aspartic acid has a carboxylic acid in its side chain. So now you have this third group that can carry, obviously in this case, a negative charge, right? When you donate the hydrogen group, the hydrogen atom, you can carry a negative charge over here. So now, in order for this amino acid to be neutral, to be in the zwitterion form, what you need is for this to carry a positive charge, and then only one of these to carry a negative charge. This carboxylic acid group over here is going to have a pKa of about 3.5. And so if you wanted to calculate the pi of this, are you going to go with pKa1, pKa2, or the pKa of the side chain? So let's just call this pKa R for like pKa of the R group. When you are dealing with acidic amino acids, in order to calculate the pi value, you want to go with the lower two pKa's. So here, I'm going to choose the one that's 2.5 and the one that's 3.5, okay? You want to go with the lower ones. So don't worry about pKa1, pKa, because I don't want you to mix up, like, is this pKa1 or is this pKa1? Like, so just go with the lower two. And if you go with the lower two, you have 2.5 plus 3.5 over 2. So that's going to equal 6 over 2, which is equal to 3. So the pi of acidic amino acids is 3. And because we know that physiologic pH is 7.5, about 7.4, which is dramatically more than 3, the surrounding environment is going to act as a base, and the amino acids are going to act as acids. So the amino acids will donate protons to the surrounding environment, and that is why they will carry a negative charge. Now let's move on to the basic amino acids. We'll discuss this very quickly since you know the concept now. And then after that, we'll move on to R/S absolute configuration. And I think we'll just discuss that. We'll discuss R and S absolute configuration, D and L classification, and then we'll call it a day. So for the basic amino acids, you can quickly draw out the structure of lysine. So you have 1, 2, 3, 4, okay? And then here you have NH3. And what you'll have here is the side chain over here will have a pKa of about 9, or let's say like 9.5. And when you want to calculate the pKa over here, instead of taking the lower two for the acidic amino acids, we said take the lower two. For the basic ones, take the higher two. So here you'll take 9 plus like 9.5, or like 9 plus 10, because it's again, these are like roughly what they are. You don't need to know the exact value on test day. So like let's say 9 plus 10 is going to equal 19. So the pKa will be about 9.5, which is significantly more than 7.4, which is the pKa, which is the physiologic pH, and that's why the physiologic pH in this case will act as the acid because it's a lower value than 9.5, thus donating hydrogen atoms to the amino acid, so it carries a positive charge. One thing I forgot to mention when we discussed the basic amino acids is that the one way they test you about the basic amino acids on test day is when it comes to histones. So remember, histones are the proteins that are involved in the packaging of DNA within the cell. And because DNA carries an overall negative charge because of the phosphate groups in its backbone, it interacts with these histone proteins. And the histone proteins are rich in basic amino acids, lysine and arginine, because that way what ends up happening is the positively charged amino acids interact with the negatively charged DNA, and you can tightly pack the DNA into what's called a nucleosome. But more on that in my biochemistry playlist. I go in depth on that. Okay, this value, 9.5, which is your PI value, is when your basic amino acids are going to exist in the form of a zwitterion. How can I have them exist in the form of a zwitterion? I'm going to have a negative charge over here, right? And then only one of these is going to carry a positive charge, right? So let's say this one carried a positive charge, this one's still neutral, right? So like still NH2. Now you have an overall charge of 0. Okay, now we're going to move on to assigning absolute configuration to amino acids. And I'm actually not going to go over specific examples because I recognize that this video has kind of gone on for a while. So we'll have a ton of examples in separate videos. Here, I'm just going to give you the general rules for R and S absolute configuration and how they relate to D/L classification in the context of amino acids. So obviously, when it comes to amino acids, the majority of them are chiral. In fact, all of them except one are chiral. Okay, and in order for something to be chiral, all you need is a carbon atom with 4 unique groups attached to it, and that is indeed what we have. So you have the carboxyl group, the amino group, the R group, and then the hydrogen group that's implicit over there. Of course, glycine's R group or side chain is a hydrogen, so glycine would look like this, as we discussed earlier. So it is achiral. So it's not going to be R or L, R or S or D/L. So with that out of the way, the remainder of the amino acids are going to have an absolute configuration. So let's just take alanine, for example. So this is CH3, the R group, and this would be hydrogen, right? So this, again, I'm not going to go into like how you assign the R/S absolute configuration because I do have those when I discuss in my videos on organic chemistry and some of my videos in the playlist on biochemistry. But what I did want to tell you is, like, let's assume this was R-alanine. And then they ask you, what is the D/L classification? Well, you should know that for all amino acids, with one exception, as we'll see in a bit, and of course glycine, we already discussed that, so don't worry about glycine, but all amino acids with one more exception, when there are, they are going to be D. Their classification is going to be D. So if you have R-alanine, you have D-alanine. Okay? If you have R-serine, you have D-serine. If you have R-valine, you have D-valine, and so on. All of them, with one exception, which is cysteine. Okay? So if you had R-cysteine, let's draw cysteine over here. Okay, so this is the structure of cysteine. If this were R-cysteine, that's just R-C-Y-S, it would be L-cysteine, okay? It's very important. This is the only exception, okay? All the other amino acids, you follow this rule. So let's say, let me write these rules out over here. So all R amino acids are D amino acids, okay? And vice versa, of course. So all S amino acids are L amino acids, okay? With the exception over here is going to be that except cysteine. And of course, I don't need to mention glycine because it's achiral. Except cysteine, when it is R equals, so R-cysteine equals L-cysteine. Now, one more rule that I want to include over here, all very, very important, okay? All biologically biologically, I spelled that wrong, but whatever. Biologically active amino acids are L amino acids. So for the purpose of the MCAT, whenever you see an amino acid that was incorporated into a protein in like human body or animal or whatever it may be, it's gonna be an L amino acid, okay? So if you have alanine, it's gonna be L-alanine, which is gonna correspond to S-alanine, right? S-alanine, L-alanine, okay? S-serine, L-serine, that's the one that's biologically active. If you have D-serine, biologically inactive, all D amino acids are R amino acids. The exception is that cysteine, the biologically active one, is gonna be R-cysteine, right? Because we said biologically active ones are necessarily L, and because cysteine is the exception, where if you have L-cysteine, you actually have R-cysteine instead of S-cysteine. Hope that's clear. I think that's all I wanted to cover. Make sure you memorize these two rules with the exception right here very well. Yeah, so I think this video has gone on for quite a while, and I claimed in the beginning of the video that this is everything you need to know about amino acids, and I think it pretty much is. Now, the only thing is that obviously you have to relate amino acids to like the different lab techniques. We're not going to discuss the lab techniques in this video. We will discuss them in future videos, and I actually have a lab techniques playlist. I actually even have a biochemistry playlist, but I wanted to teach it in a different way because different people learn in different ways, and just from my experience, like as I do more iterations of these videos, I tend to produce better videos. So that's why I like to teach the same concept in different ways. I feel like as I gain more knowledge, I can teach the material better. So you will find in the description box the link to the Anki cards for amino acids. You'll find the practice sheet in the description box and the biochemistry and the lab techniques playlists, okay? With that, there is one more thing that I didn't discuss related to amino acids, just so you know. Titration curves, and I have a separate video on titration curves, so I will include that in the description box. But with that being said, I think like just by having this material and really mastering it, it'll yield you at least a few points, if not several points, on every single full length that you take and on test day. So definitely master these concepts. Also share these videos with your friends. Not only is it going to help them out on the MCAT, but it really helps this small channel grow, and I would really, really appreciate it.